This is TWIS, This Week in Science, episode number 601, recorded on Wednesday, January 11th, 2016. Back to grinding science. Hey everyone, I'm Dr. Kiki, and tonight we are going to fill your heads with marital strife, spider webs, and superheroes. But first...
Disclaimer, disclaimer, disclaimer. A funny thing happened on the way to the modern world. Evolution. We are here as the result of evolution. It is the process of life that led up to the natural world. But evolution is not just the thing that happened, and it is not just the process of natural things. Evolution is taking place in the modern world as well. Evolution is everywhere. In our societies, cultures, and economies, evolution plays out in our technology, art, and engineering. Evolution is the process of trial and error, the process of learning and remembering. Evolution is life in all of its endeavors. And science is at once the study of evolution and the act of evolving. The current state of our evolution depends more on science than it does natural selection, yet natural selection is the prototype upon which science was built. Make no mistake, we are evolving right now, faster than ever before, and nowhere else is this active evolving now talked about more than right here on This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Science to Kiki and Blair.
Ooh, Kiki, you're muted.
Oh, and a good science to you, Justin, Blair, and everyone out there. Let me hope that in this second week of January that none of us are muted more than we need to be. I mean, maybe some people need to have their Twitter removed, but
the science shall not be silenced.
The science shall not be scienced. Science and reason for the win. Here we go. We're going to join the rest of the world in celebrating a little bit of science this week. We got so much science happening. And you know, we still have a couple of these This Week in Science 2017 calendars available. So please check out TWIS.org in order to find out more about the 2017 calendar for TWIS that Blair made all by herself with Patrick Hartnett's help. And next week, Thursday, the 19th, we are going to be in San Francisco, California, at the Cal Academy of Sciences Nightlife SF Sketchfest event. We're going to be doing our show in front of you. Yes. If you can make it, I hope you can make it. That would be really awesome. Bring calendars. We'll sign them in person. How does that sound? And is there anything else? Oh, yeah. This weekend, I think it is National Citizen Science Day on Saturday. So everybody, I hope you get out and get sciencing. Be a citizen who sciences this Saturday. Find something fun to do. Lots of fun stuff going on in the world. And are you ready? It's time for a fun, action-packed show. We have so much stuff ahead. I have got stories about the appendix, baboon speech, and scientist spider wannabes.
Ooh.
Yeah. What do you have, Justin?
I've got more retro than retroviruses, one less blind mouse again, and how aging is all in your head.
Yeah, I see what you did there. Blair, what do you have?
Oh, I have avian marital bliss. I have avian divorce. And I have smelly armpits.
Well,
I
we
don't
all
need
know
it.
that you
TMI.
probably
TMI there, Blair.
shower a lot, so.
TMI.
Very.
Right. Hardy, 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 har. Okay, it is time for us to jump right in. Let's do it. I have a wonderful story. A wonderful, wonderful story that takes us back. But doesn't take us back at all because it happened just now, recently. A bunch
Just
of
happened, yeah.
it just happened. A bunch of researchers from all sorts of different places came together to publish a paper in Nature this week titled Asgard Archaea Illuminate the Origin of Eukaryotic Cellular Complexity. Now, the main researchers from Uppsala University in the Netherlands, Sweden, in Uppsala in Sweden, hence the Asgard reference in the title of the paper. What all these researchers have done is a genetic analysis of various samples that were taken from underwater environments. And so they took, I think, 6 or 7 different samples, 7 geographically separated sites. And one of them is called Loki's Castle. Another one, Yellowstone National Park. Another one, Aarhus Bay. Then an aquifer near Colorado River, Radiata Pool, Takatomi Island Vent, and the White Oak River Estuary. Then, instead of like normal biologists wanting to look at microbes, putting them in a dish and seeing what they could get to live, oh no, they didn't care what they could get to live. They just looked at their genes. They took the DNA from all the samples and they just sequenced them and they ran them through machines until they could figure out what genes were there. And they said, okay, based on the genes that we've got here, who lives in these places? And now this story goes back to another story that we reported on back in 2015 about an archaebacteria called Loki. Now, Loki, again, named by the same team, Loki archaeota is an archaeal clade that was previously shown to be more closely related to eukaryotes than any other prokaryotic lineage. So eukaryotes, multicellular organisms with very complex cellular structures versus prokaryotes, which are unicellular organisms, bacteria, where things potentially started, right? And so evolutionary idea is that we started with bacteria, single-celled organisms, and at some point there was a symbiotic merger between organisms that allowed more complexity to evolve. And Loki was the first discovery that really kind of hinted that this hypothesis might be true, because when they looked at Loki's genome sequence, they found a whole bunch of genes that they were like, hey, there's genes for cytoskeletal structure in here. There's genes for all sorts of things that are usually, we think of as only eukaryotic. And so it was kind of like, okay, Loki must be one of the in-between species, right? It must be one of the steps between bacteria to more complex eukaryotic life, or related to, right? It's the closest living relative to those steps.
Yeah.
Yes.
Well, and maybe I'm conflating with this another story, but was Loki also possibly being looked at as like a eukaryote, maybe that had gone backwards, like had evolved, and then it was like, forget this.
That's one of the hypotheses. I mean, that's a possibility, but the easiest answer here is that Loki is an archaebacteria. It fits genomically within the archaea family, and that it's an archaebacteria that just has more complex structure. So it's kind of a wing of its own, it's this branch on the tree coming out on its own. And so hadn't seen any other organisms similar since or before, but this paper announces an entire group.
Mm-hmm.
This is an entire group related to the Loki archaeota, and they're called the Asgard archaeota. The Asgardians, the superphylum. It's discovered through metagenomics. And it has a number of individuals. Thor is one that they named.
I'm
Thor.
sensing a theme.
At some point, I'm going to feel like I'm learning more about Norse mythology.
Yeah, we're dealing with Norse mythology here. Yeah.
Is it Norse mythology or comic book fans?
Right. No, this is Norse mythology. Swedish researchers.
It's hard to tell the difference at this point.
Yeah.
Yes.
Yeah. So we have Loki, we have Thor, we have Odin, and we have Heimdall.
Okay.
Heimdall.
If they had football teams, they'd all be named after the gods, and it'd just be it's like their one theme that they've got.
Yeah,
I get it.
So it's
keep running with
it. So it's kind of neat, they haven't cultured any of these in the lab. These are strictly found based on genes, and this Asgard clade that they have put together, they have so defined because they all contain genes that had heretofore been only defined as eukaryotic. And so now we find that potentially the archaebacteria, or maybe other bacteria, were setting themselves up to evolve. That complexity and this genetic complexity was evolving well before eukaryotes came onto the scene. And so does this have to do with symbiote? Is this the symbiosis of these different bacteria coming together to become superorganisms? Is that what's going on here? I don't know. But there's a lot of different questions that are going to come from this, and it's very exciting research that we're finding. This is potentially going to lead us in the direction of understanding how we went from unicellular to multicellular organisms. Very neat research. And they don't have to grow anything in dishes.
Yeah, machines are taking bacteria's jobs now.
Yeah, nothing has to be alive. It can all be dead. Yeah, so it's a very cool study. Yeah. And then moving on from this illumination of eukaryotic cellular complexity that's been given to the Norse gods. From there, I'm going to take us to spiders.
Yes.
Because why not? We love the spiders. We love the spiders because they have spider webs. No. For a really long time, researchers have wanted to spin spider silk. Spider silk.
Yeah.
It's stretchy. It's strong.
Very strong.
Oh, it's amazing substance. I mean, we 10 years ago, maybe even more now at this point, Justin, we reported on the spider goat where they're genetically modifying. Yeah, they're
spider goat. Spider goat.
Right. They're creating goats to create spider proteins in their milk so that scientists and manufacturers would potentially have a very large source of spider proteins to make this silk. But they have not been able to actually chemically make all those. They have the proteins. Great. They can produce them in the goats. That's awesome. But they can't get them to mix together to create the nice threads. It's been a problem. And so finally, a Swedish group, Swedish University of Agricultural Sciences, have reported that they can produce a kilometer-long thread.
What?
That is a long thread.
That's
kilometer-long thread.
So for those out there who aren't familiar with kilometers, 1 kilometer is about the same as 1,000 meters.
Yeah.
So you put all the 1,000 meters together, that's very long.
Yeah, probably equal to about how far I walked today. Not out walking in the snow today. So they're able to take a highly concentrated solution of the spider silk proteins in a syringe, and then they pump it through a really tiny glass capillary, which it's just a tiny glass tube, basically. And it has a tip the size of like 10 to 30 millimeters, really, really tiny. And then they submerge the tip into a low pH aqueous collection bath. And the fibers come out, can be taken from the bath and rolled up onto frames. And so it's a very exciting development here that they're able to actually create something that resembles real spider silk. And so what was important here was taking the actual spider gland and looking at how the spider gland puts together the spider proteins. Because in the spider gland itself, there's not just a bunch of it's not like a spool of thread in there, right? The spider silk gland is not just a spool
I always
of
thought
thread.
it was.
Not just a spool of thread. That's not what's going on.
I pictured it.
It's
Tiny
like
little
it's a
bobbin in there.
liquid. It's a liquid protein soup.
Right.
And the proteins don't congeal and turn into that thread until it's exiting the gland. And it has to do with pH gradients. And so there's just a really well-regulated pH gradient between the inside and the outside of the gland that affects the proteins, so that basically the fiber forms rapidly in a very defined place and nowhere else.
It's kind of like it reminds me of, I was going to say it's a backwards glue gun, but it's kind of more like a 3D printer.
A little bit more like a 3D printer.
Yeah. Although that starts with a spool also, but it's more like a backwards glue gun. So it starts as liquid and then it comes out as a salt.
Yeah, so the pH is, I think, very interesting because it is able to, like, the high pH probably denatures the proteins a little bit, and then as it exits, the proteins are reassembled. You know, they start twisting back into their proper tertiary form, their proper formation after they've been kind of spread out and open so they couldn't attach to each other, and then they attach. And so if they're able to wind up and attach, then you have a spider. And so the researcher says, to our surprise, the artificial protein is as water soluble as the natural spider silk proteins, which means that it's possible to keep the proteins soluble at extreme concentrations, which is fantastic. And this is the first successful example of biomimetic spider silk spinning.
Woohoo!
Yes, and so this could lead to spider silk shirts.
That's great.
Yeah.
So actually, I want to tease too. There was so much science news this week that I didn't get to bring it all this week.
Huge week, yes.
I had to save something for next week that is about making that spider silk t-shirt even cooler.
Oh, it's going to have lasers.
You guessed it. I don't have to do it next week. No. It's something great, though. You guys will have to tune in next week.
It's going to glow in the dark and have lasers. It's going to be its own DJ nightclub t-shirt.
You're going to ruin the surprise.
Sorry, you guys. I didn't.
And actually, it should cut down on barroom violence.
Yeah.
You know, when people get really mad and they're gonna like rip their shirt off to have oh yeah, buddy,
I can't rip it. It's spider silk. It won't rip.
Never mind. Never mind. I'll buy you a beer.
Yeah. It's actually, this will be more about when you're in the club and somebody accidentally gets sick all over you.
Ew.
It'll be helpful for that.
Ew.
Oh. That also doesn't happen.
That doesn't happen to me because I don't go to clubs anymore.
Ding, ding, ding.
No, but there's going to be like, you know, you get your shirt caught in the elevator or something, and then instead of being able to rip out, there'll be somebody who's like, yes, the dangers of wearing spider silk. It will kill you if you get caught on anything.
You just hang there. Your shirt would never rip. You just
Yep.
hanging out.
Yeah.
Your shirt's not ripping. Everything's great. Okay. Anyway, we've taken this tangent far enough. This is This Week in Science for those of you watching and not able to see the lower thirds. We don't have any this week. And for those of you listening, if you just tuned in, that is who we are. This Week in Science. It's Justin's turn right now. What you got, man?
What did I say I was going to bring? Retinal degeneration. There's apparently a mostly hereditary disease characterized by the death of the photoreceptors in the eyeball, light sensitive neurons in the eye, which leads to blindness. This is You know, this is when you're talking about blind people. This is not the somebody who forgot to put on the safety glasses and was working the wood chipper. This is like sort of born blind or born and then lost sight shortly after. There have been attempts to do retinal transplants to combat the disease. There's been some transplanting of graft receptors to the host that had some integration, but Ccience has never successfully transplanted photoreceptors that functionally connect to host cells and send visual signals to the retina in the brain. So we wait for that day for science to progress to the point where I will have a story to tell you. Oh, wait, what?
What?
No, a team led by Masayo Takahashi just did it. Boom, science just happened. This is actually a continuation of the story I reported late last year. But what's wild is that they had initially done this test with putting these little points of luminescence where they did this 3D retinal sheets derived from stem cells. And because they sort of put these little glowy bits on it at the end of the photoreceptors, they were able to tell that the thing actually went back and connected to the right parts of the brain. So they're like, wow, we have not only the thing that can pick up signal, but it's actually connecting itself back to where signal is read. So what they did in this was they used a mouse model that was at end stage retinal degeneration, which the outer nuclear layer of the retina is completely missing. The outer layer is completely gone. This is they used as their starting point. They used the worst possible starting point. This is the end of the road. This is the end of the degeneration. Worst possible starting point, and they applied their method to it. Wanted to see what they could actually do. So they learning tasks. They had these learning tasks that they were using with light and sound for mice that had perfectly normal vision, and they trained them to do this sort of little task, right? So then they went back to those that had the complete degeneration model where they had no vision, and they tried to do this learning method with them to teach them this sort of behavioral task. And of course, they had no result. Then they tried it on those that had received the transplant, and it worked. They were seeing stuff. It may only be certain amounts of light. We can't really tell, but we do know that vision with light is there. This is now going to go into a human trial.
Yes.
That's awesome.
This is, yeah.
One of my predictions is already going to come true.
3 not blind mice. 3
We are planning to
not
proceed
blind
to
mice.
clinical trials in humans after a few more necessary studies. And they're going to use, of course, human-derived retinal tissue. They're in a few mouse models, trying to make the whole thing go all the way across. They say clinical trials are the only way to determine how many new connections are needed for a person to be able to see again. So they're gonna keep working this direction. But this is the blind man shall see, science curing blindness. I mean, this is not through, there have been attempts. There was some weird one that actually worked where they would implant a tooth. Remember that one?
Yeah.
They would implant a tooth. I mean, there have been all sorts of implants and attempts and things, but this is, I've seen or heard since we've been doing the show at restoring natural vision without, with the natural eye, I guess. So,
wow.
Progresses.
This is very exciting. I can't wait to find out what's going to happen with the human trials and, the hope, very, very, to put the realistic slant on it. We get excited about stuff like this, but until it does pass the human trials, we don't know if this is something that will work in humans.
Right. It is mice. Giant asterisk mice.
Yeah, mice are different from people.
Mm-hmm.
And maybe the mouse eye and the system is similar enough that it'll, all of this graft to host stuff is going to work easily. But,
So, yeah.
That's the
so there's
hope.
there's the chance that we're just less regenerative,
Yeah.
that our stem cells are going to be like, what do you want me to do? I have no idea.
Exactly. But I mean, when you're using, the whole induced pluripotent stem cell tissue model, I mean, in the pluripotent stem cells, the induced pluripotent stem cells, they're at a young stage. They've been taken back to this early stage of tissue growth. And so they've kind of their clock has been taken back, and they want to grow and they want to do things. So maybe, there have been things that have worked in humans with iPSCs, and this could be amazing. Could be amazing.
I say get your hopes up.
My hopes are up already. It was in my predictions for 2017. Was a human trial.
Hopes up high, people. This is the type of hope you've been waiting for.
Blair's I want to get rid of my color blindness.
Yeah.
Well, no, but I mean, like, if
that's different. Yeah, that's totally different. No, I was responding to Rob the Stressed in the chat room, who said, I want to see in more colors and also ultraviolet. And so I said, I want to see in normal colors.
That's right.
And I'm just sort of picturing this from if I was blind. I would be exceedingly encouraged by this, and I would have absolutely insanely high hopes.
I will say, I just want to mention too, though, that when cochlear implants happened, when that was a new scientific breakthrough, there were individuals that wanted them, and there were individuals that didn't want them, and there were individuals that got them and then had them taken out. And I think that's an interesting point to remember too, is that if you are born blind, you've been blind your entire life.
But this is for people with degenerative disease.
Right.
So this is going to be
absolutely.
This is people who have gone blind.
And that's going to be huge, because if you've learned this world with sight, losing it has got to be the worst, like the literal worst. But that does mean if we figure this out down the line, there could potentially be something for people who are born blind. And that's just something that I do think about, is that when you consider altering people's entire sensory experience for their entire life, there's people who are going to want it, and there's people who aren't.
Yeah, absolutely. And so, like, Twit Refugee is saying there's no absolute brain structure that makes sight, so they'd still be functionally blind. And I don't think that is correct for this particular instance. So this is for retinal transplantation, specifically for people who have had retinal degeneration, so that they had sight, the retina of the eye degenerated, and that's the light collecting part of the eye. And so if you can replace that tissue so that the retina comes back, the brain is all still there. All the systems are still there. So if you can put put that little part of the camera back in, then you should be able to get the picture back.
Absolutely.
Yeah. So it should theoretically work, because these are people who could see and then lost it. Yeah. Very exciting news, Justin. Oh my God.
The future is now.
Future is, it's 2017.
It's
happening.
It's now. No, it's now. No, it's now. No, it's later.
I wonder if we'll have. I wonder if we will have trials start this year and results back by early 2018.
That's what I need for my predictions to be accurate.
Oh, that would be so exciting. All right. Well, if you just tuned in, again, this is This Week in Science, and we're talking about science. And you know what time it is in the This Week in Science show program? Can you guess? You're going to guess, Justin? You can guess.
I think it's time for Blair's Animal Corner.
Bugs are creatures, great and small. Biped, millipede, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I have avian relationships. The good and the bad. Yes. Let's start with what may sound at first like the good, and that is common terns. Common terns are birds they look a lot like fancy seagulls, but they are extremely, extremely monogamous. New research from Max Planck Institute in Denmark and also several different institutes in Germany. They all did this combined study of terns. And they were specifically looking at the marital dynamics, we could call them, because they are so monogamous. They find each other pretty much the second they're sexually mature, and they stay together for the most part till they die. They did a 17-year study. They looked at a colony of over 1,300 individually identified birds nesting on 6 artificial islands in Germany. And what they found was that these terns were staying together no matter what. No matter what. Whether they were a good genetic match, whether they had a lot of babies or not, whether they were either of them were fit to be good partners, they stayed together no matter what.
We stayed together for the kids.
Right. But what if you're not having any kids?
We weren't happy at all.
What if there's not any kids though? Still staying together. Why? Usually in situations where there's intense monogamy, there's some sort of benefit that couple gains from it. So either they pick a really good individual, or if the pair is not a good fit for each other, there will be what we call divorce, which I'll talk about in a little bit.
Okay, because I was going to say there, I don't know what the difference between intense monogamy, extreme monogamy, or just monogamy.
The reason I said intense monogamy,
you're monogamous,
is that
you're just not monogamous. You're not like, it's not a scale, really.
There is a scale, because there are animals that pair bond but have extramarital relationships. There are pairs that break up after 3 or 4 years. Penguins are the perfect example. People call penguins monogamous animals, but they break up after 2 or 3 years all the time.
But then they're monogamous with another penguin, right?
Yes. This is lifelong monogamy. Yeah. And so these guys, they were staying together even if they were a bad match for each other, even if they weren't having babies. And they were not having more babies on the whole, on average. You would expect overall they'd be having more babies. They would be more productive pairs. It would average out that way, because in that way, monogamy would still be beneficial. But that's not the case with these terns. What they found is that monogamous terns, even if they do, in the extreme case, break up, finding a new partner does not result in more fledglings either.
And how do you find a new partner if everybody's extremely intensely monogamous?
Exactly the hypothesis now, is that this is all about availability. It's kind of the greatest catch-22. The chances of finding a partner that's your age is slim to none, because everybody pairs up the second you become sexually mature.
Wow.
So there are no single terns.
Unless there's something really wrong with them, which is also why I'm afraid to ever date. Ever.
One could die is the other.
Which is why I would never date again.
Kiki, you're muted. I can't hear you.
It's like being a single female in San Francisco.
Bingo! You got it. Yeah, so this is the greatest catch-22, because everyone's in a relationship, because if they became single, everyone's in a relationship, so they couldn't find anybody.
Yeah. That's the way it is in human society, too. Have you not noticed
Absolutely. Absolutely. So they just find somebody as soon as they become sexually mature, and then after that, it's done. They've sealed their fate. So pair bond length is not what drives the tern's reproductive performance. Instead, they found that age and differences in breeding quality among individuals are important. And since the terns pair up as soon as they become sexually mature, we see all these same age pairs in the colony. So you know what you have, not what you may get. So they stay with what they have.
Okay, so
that's a strategy.
Yeah.
Right. As a strategy, what do you think about this? So we're sort of like, I don't know how many our terns are in a what is it? A flock of terns?
Yeah,
I
so
don't know
for
what it
example,
is.
this
Gaggle of
study
terns?
looked at 1,300 individuals on 6
Okay.
different islands. So,
Okay,
but
so-
terns are seafaring birds, so they can go pretty far. But it also worth mentioning, this is a subarctic colony, and these guys are Arctic animals, so the resources are pretty scarce.
Right, but what I'm getting at kind of is like, is there sort of a different advantage in a monogamy gene pool than there is of one that's sort of mixing about? And I guess in a way, when you've got a population of 1,300, the less inbreeding or the less crossbreeding you do, the less inbreeding that'll take place, right? You can sort of create slightly more distinct gene pools that can go out a little further each time and then reconnect later down the road than if there's a lot of crossbreeding going on. So this is a strategy. I think
that's
it's pretty
true.
interesting.
Yeah, but I will mention also there was another great study this week. There was so much science this week about how chimpanzees can absolutely tell who they're related to and who has the most genetic diversity from them. And this is something we've talked a lot about on the show. Animals generally know. Animals generally know who they share too much genetics with, and nature has a way of working that out, in that a lot of the time that'll result in infertility. So I think it's more about the fact that they just don't have time to dilly-dally. They don't have resources to dilly-dally. I don't know if also
Gotta make it count.
Is that
got
a euphemism now?
they dilly-dally. They don't have the resources, the energy, I don't know, the time, let's say, to compete with each other every year. Maybe this is something where they'd rather, as Arctic species, the first year find somebody and then go, okay, I'm letting myself go now. I don't have to compete for a pair every single year. I don't know. I think it's something to look at to see if there's something about resources, because again, we brought up penguins, Antarctic animal.
All I can say is you're in a resource poor environment where people where the animals have to fight between, it's a struggle to get what they need. If you've got a partner who's going to help you, that's one less thing you got to worry about. So it seems like in that kind of ecosystem, it's a good strategy.
Yeah. Absolutely.
You might not have any kids, but as long as you're not dying, I've got a partner.
Absolutely.
It's helping me out.
Yeah. A poor partner is better than no partner, I think, is ultimately what the scientists were looking at there in this case. Yeah, I would not give that life advice to humans necessarily, but if you're an Arctic tern, that might be the way to go.
And then moving into your songbird story, this is the opposite.
The opposite, divorce. Songbirds will undergo divorce, which is exactly what it sounds like. They're a monogamous pair that break up. And their divorce rates, coming out of a University of Washington study, their divorce rates are directly related to suburban sprawl. So songbirds are avoiders. What that means is they don't want to be up in humans' business. There's other animals that maybe will nest in a bird feeder you put in your backyard, or an example that they put in the story that I thought was pretty funny was a wreath on your door. There are certain birds that live in urbanized areas that will take advantage of every possible nesting opportunity, no matter how close to humans. But a lot of songbirds are avoiders. They will avoid direct contact with humans. And so as suburban sprawl happens, as people move further and further out into the country, it forces these avoiders to also flee. And this makes them do things that natural selection otherwise wouldn't have them do. And one of them is divorce. So as they flee, they may flee away from each other as well as from the humans, and then they have to find a new partner. And when they do that, a lot of the time they miss an entire breeding season. So these birds, they're traveling uncharacteristically far, about 1.5 football fields on average from their current home when the suburban sprawl takes over. That's a pretty far span for them to then re-find a new home. And So this means they have to find new resources in their new space. They have to find a new mate, and they have to set up a new home base. So in the effort of doing all of that, they will often miss breeding seasons. And so that can be really devastating to songbird colony sizes, and it can be really devastating to have that divorce rate as well, because having to find, this is right along with what we were just talking about, that's a whole new energetic cost that they wouldn't have had to do that season multiple times in their life, however many times. So whereas exploiters, those are animals like juncos that would breed in your backyard.
I love
Their
dark-eyed
chances-
juncos. They're
Yeah,
so cute.
They're
And-
so cute.
But it's true. They are all over the place. You will see them in cities. You see them all over the West Coast of the United States. They are in the fall and winter. They are prolific.
Absolutely. Yeah. And so these guys don't mind the urban sprawl. In fact, it doesn't really hurt or help them and their life cycle and their fecundity, their ability to have babies, or their breeding success. But for these avoiders, it's hugely detrimental. And this is about a 10-year study. So it's pretty reasonable study size. And this is a brand new impact. It's something that probably we could have guessed that it would affect animals, but not exactly in this way, that it's habitually going to make them miss entire breeding seasons.
Yeah, I think I never expected individuals to get run off in that way.
Mhm,
You know,
mhm.
you think of it in terms of groups of birds. There's a flock of birds. There's a population of birds. They all kind of know where to go to meet each other and to hang out. And how can one bird or another bird kind of get lost from the system?
Absolutely.
And so that's interesting that human behavior, human activities can really have a dramatic influence on some birds, but not others.
Yeah.
Because of this. Yeah.
Absolutely. And one main characteristic of suburban sprawl is that you still have a fair amount of wild space around, but it's fragmented.
Mhm.
So you might have a thoroughfare of homes right down the middle of a space that used to be wild. And that, for a flighty animal, is going to mean that they're not comfortable anywhere near that space because they'll need a large home base. So suburban sprawl, I'm not surprised to hear it's affecting animals and, of all things, birds, but this is definitely something that is going to be looked at closer as we continue to look at the effects of humans on animals and birds in particular.
Yeah.
Divorce.
Yeah.
It's
Yes.
our fault.
We're making birds get divorced.
It's all my fault. I'm sorry.
People are splitting up very happily married bird couples. Oh,
But they said the divorce wasn't my fault.
people. Oh, people.
love
what are we
nature.
doing?
Stay away.
Yeah, if you love
Yep.
nature, get out.
Yep, that's what we learn time and time again.
Oh my goodness. Yes. All right. Well, you know what? Don't get out of this show. Don't go anywhere. We have reached the end of the first part of the show and we have more to come after the break here. So stay tuned for more This Week in Science. Hey, everybody, just to let you know, once again, we do have a few of these calendars left, so please get them while they're hot because there will not be any more once they are gone. This is the 2017 This Week in Science Blair's Animal Corner calendar, hand-arted by Blair. Lots of science holidays that you can follow along throughout the year. Know when to celebrate. Know when to celebrate. Know when to science. That's right. National Citizen Science Day is this Saturday, the 14th. You know how I know that? Because it's in the calendar here. That's how I know it. And you know what's going to happen next weekend? Squirrel Appreciation Day. That's when you send Blair lots of pictures of squirrels. Anyway, go to twist.org to be able to find out information on how to get yourself a calendar before they are all gone, because that's going to happen very soon. If you don't have a calendar yet, you got to get one. Also, San Francisco SF Sketchfest show, January 19th. Go to our Facebook page, facebook.com/thisweekinscience, to be able to find out about that event and to catch a link to the ticket sales at the Cal Academy of Sciences. Thursday, January 19th, sometime after 6 p.m., we are doing a show at the Cal Academy of Sciences SF Sketchfest nightlife event. It's going to be so much fun. We hope that we will see you there. That would be fabulous. TWIS also has merchandise that you might enjoy. So if you are interested in getting your hands on some items that are TWISy in nature, head on over to twist.org, where you will be able to click on our Zazzle store link. And the Zazzle store link will take you right on over to the Zazzle store. In the Zazzle store, you'll find our logo emblazoned over all sorts of neat products that you might enjoy having. Do you need a mammoth lumbar pillow? Well, if you do, that lumbar pillow was hand-arted by Blair. That's Blair's art right there. We also have TWIS polo shirts now. So if you are interested in having a polo shirt for any work or school dress codes that you might have to deal with, then you can now get a TWIS polo shirt. It's very exciting. iPhone cases, necklaces, wrapping paper. You know, we've got all sorts of stuff in there. Hope you'll find something that you enjoy that has art from Blair's Animal Corners or from our TWIS logo. That would be fantastic. Additionally, TWIS is supported by listeners just like you, by viewers just like you. And if you would like to support TWIS, please click on our donate button. It's on twist.org. It's a little yellowy, orangey donate button. It says donate. You click on it. It makes it really easy for you to send us money via PayPal. If you're not into PayPal, you can also click on our Patreon link. The Patreon link will take you to our Patreon page, where you can click and become a patron. Support TWIS. Become a patron on a weekly or a monthly basis. You decide how much you want to give on an ongoing basis. And we send you little tiny gifts in return. And sometimes you get things before, you get to see things before anybody else does. That happens sometimes. It really does. It really does happen. So either click on that donate button on TWIS or head on over to Patreon, patreon.com/thisweekinscience, to be able to support us that way. If you would like to support us some other way, there are many, many options there as well. Contact me via email, kirsten@thisweekinscience.com, if you'd like to chat about something, if you have an idea, or simply share TWIS with your friends. That's right. That's all you have to do. Social media. Put out the link for the most recent show. Send the twist.org link out to people you know on Facebook or on Twitter or on Instagram. Take a picture of yourself hanging out with the TWIS calendar. Post it to Instagram. Take a little video of yourself watching TWIS and post it to, I don't know, Periscope or Snapchat. Or does Snapchat do videos? I don't even know. I don't use it. Oh dear. So much social media. So much. But I know you might use it. And if you have a way that you can help get the word out about TWIS, please help us. It would be very, very helpful, because really, this is a show that is supported by our viewers, our listeners. You keep us going. You keep us making this show week after week after week. We thank you for your support. We really cannot do this without you. Thank you.
The miracles and cures all laid down in black ink, never even bothering to stop and think. The only real power that I do believe is the dollars and cents all these authors receive. If miracle wonders were held in their looks, why waste precious time and try selling their books? Why sit?
And we are back, aren't we? With more This Week in Science.
Oh, yeah, we are back, and we have more science for you for the end of this show. We've got a little bit more to go. Justin, what do you have to tell me about? What you got?
This is new research by scientists at Oxford University, and it is redefining the word retro when it comes to viruses. So retroviruses are a family of viruses that are made out of RNA, which they then can convert into DNA and insert themselves into the host's genome. And then they can stick around for a really long time. They can actually be inherited. You can sort of inherit this this virus within your genes. And so there's a history of these retroviruses in human DNA. We can sort of track them back. There's also active retroviruses that are out there causing harm, like HIV. And it's been thought that these have been with us for millions of years. But according to research, they have been with us since before the first fish walked on land, or whatever happened. 500 million years or so ago, give or take all of human history. The findings reported in Natural and Journal Nature Communications will help us understand more about the continuing arms race between viruses and their hosts, says Dr. Aris Katsarakis, Oxford University's Department of Zoology. Our inferred date of the origins of retroviruses coincides with the origins of our adaptive immunity. And thus it is likely that retroviruses have played an important role in the emergence of this key tool in vertebrate antiviral defense. As we understand the nature of the interaction between viruses and host immunity, we will be better placed to intervene in this delicately balanced arms race in order to develop novel treatments and interventions. So that's in itself an astoundingly wild and awesome find, that the sort of origin of the retrovirus is also taking place at the origin of our adaptive immunity. You know, it's one of those things that I think we just learned this last year, that infants have a huge, what do you call it? A huge amount of viruses in their guts, right? And when they're first born, and there's these viruses are there, mostly feeding upon bacteria. And they become, in a way, a filter, like a screen, like which bacteria that are in this infant are going to continue on to develop into the microbiome of this. Kind of depends on the viruses. And the viruses sort of drop off at about age 2, and the bacteria that is still there afterwards is what goes on to develop into the infant microbiome. So knowing now that, you know, these retroviruses, they've sort of traced them back in time to the invent of our immunity, our adaptive immunity system. That's huge.
Yeah, it's pretty neat to be able to start putting a retroviral tree together and start figuring out, like, okay, where did they come in? When did the retroviruses really get in there? You know, when did they, what was the origin? When did it happen? And it's so neat to see that, you know, it was a long time ago. Retroviruses have been infecting us for a very long time.
Since we were fish. I mean, this is, that's about as far back as I can.
We were floating around in the water with the viruses, and they're like, all right, chomp.
But you guys, viruses aren't alive.
Not in the way we are.
Yeah, life.
I mean, cheeky, because I always thought that was dumb. Of course they're alive. They evolve.
No, this is it's open for debate. It's one of the gray areas, isn't it?
Science is just full of gray areas.
All sorts. That's what I love about it. Love it. Like another one, oh my goodness, not so gray, but full of sound. Full of lots of wonderful sound. I don't know if I can make you hear this.
It sounded like a dog barking in the distance.
You know what that is? That's a baboon saying wahoo.
Ew.
Wow.
This is right along the lines of "I love you."
No, she's
no.
saying-
What was it? Will you marry me? That's what it was.
Mhm. Yes. So that story in which we talked about the finding that the macaque larynxes are totally capable of making human sounds, right? This is one of the first studies that's really that last year we're like, okay, this is something that's come out where forever we've just said, okay, the monkey larynx, this early primate larynx is too low or too high to be able to create the right sound compared to humans. And so other researchers were like, well, maybe if we look at humans, that there are some people, there's variation in the placement of the larynx and how it works, and babies are still able to make these sounds. So
and also it's sort of like the whole idea of it too is like the reason this ape doesn't talk is just because it can't. It tried, it gave up a long time ago.
Yeah.
Just because they couldn't articulate themselves properly. They just gave up on the whole vocal communication.
Well, it's more like,
Yeah,
well, they're not smart enough. Wait, actually, it turns out that they are. Oh, so they don't have the mechanics. It's not possible. Oh, it turns out. Yep. Okay.
And now
they
a
just
new
have nothing to
study,
say.
and now they have a lot to say, just not like us, but very similar to us, actually. New study reports that Guinea baboons, these monkeys inhabit forests and savanna of West Africa, can make 5 vowel-like sounds similar to those that are used by humans. And in fact, in some human languages, we only use about 3 vowel sounds. So these baboons are capable of making more human-like vowel sounds than some human languages even use, which is fascinating. Yeah, so I think this just adds to the work that on the Japanese macaques about the anatomy being capable. Now, researchers, primatologist Joel Fagot, primatologist at Aix-en-Marseille University in France, and is the author of this PLOS-1 study. So you can actually view this study. It's open to anyone because it's a public library of science publication. He looked at 1,400 or so vocalizations of 15 Guinea baboons that are living at a primate center in France, and looked at their repertoire. And so they had grunts and barks and copulation calls made by females, distress calls known as yaks, and wahoos, wahoo, and which is a long distance contact that's most often made by males. And so they produce 5 distinct sounds, at least, that correspond to the International Phonetic Alphabet.
So they just lack consonants.
Vowels. They're not consonants. They're vowels.
No, they just lack. They lack consonants. That's the only thing keeping them from talking now.
Exactly. Yeah. This is it. This is the new defining feature.
If they figure out how to go
consonants.
It's all over. It's Planet of the Apes. Here we are.
That's right. And so it turns out, like in this wahoo sound, this wahoo that I played just a moment ago, this is a combination of 2 vowel sounds being used. So this is it's a word. It's using a complex combination of sounds for communications. And so they have the building blocks. Doesn't necessarily mean they have rules and structure for language, but this is very exciting.
Like the
this actually is a gray area, because it's one of those things that we keep saying animals don't have language, animals don't have the capability for language, and it's turning out to all just be gray. There's a very fine difference between what we do and what other species do, and we may find out eventually that there's no difference at all.
Could be.
Maybe. Yeah, or it could just be like maybe they literally just can't figure out a consonant. Maybe they can't make a T. Maybe if we as the species,
the study
could only roll our R's,
right?
And that's all we could do.
Like,
we could only communicate so much by continually just rolling R's.
But that makes me wonder too. Have we gained a giant vocabulary at the detriment of our ability to read other ways of communicating.
Yes, but we still use body language.
Yes.
don't think so.
Yes, but
I don't think so.
Do you think that just as we can say that their verbal communication is not as complex, that their body language communication is more complex than ours? I would say yes.
But we don't know.
I would say no.
We haven't looked
I would
at
say
it.
no.
We don't.
We
know.
haven't looked
Well, I
at
mean,
it,
I
but I
wouldn't-
think it's an interesting perspective.
But I doubt it.
Consider.
I doubt it. I'll go with Justin on this, and I'll say I
and
doubt.
I don't mean to, I guess I was saying I was doubting something else though. I was not doubting that they have communication that's body language that they're picking up amongst each other that we can't read from them as well. But I wouldn't say that we don't read it from each other as well. And from and if you meant from reading other species generally, I mean, I feel like I'm much more able to recognize an erratic driver or a driver who isn't slowing down than say a squirrel, right? Like, but a squirrel might have a little better beat on when that rat is coming too close to its nest or looks like it's up to no good or whatever it is, you know.
Yeah, I've run over way more kamikaze squirrels than I have had auto accidents with erratic drivers.
Yeah,
Sure.
Right? So I mean, like, I think you do learn to pick up on read a lot of things within your surroundings, within those things that you're interacting with, and so
another aspect
they're having more communication between each other than we're picking up on. But do they have more communication than we have with our surroundings? I doubt it.
Yeah. And then another aspect of the study, they dissected the vocal tract of 2 baboons that died in this location. And they found, I don't know how nobody ever saw this before. They have the same muscles in their tongues that we do. So talking about consonants, they very likely should be able to make those sounds.
So there we go.
They have the similar muscles in their tongues.
Let the gray area lengthen.
Let the gray area go further. All right, Justin, what else do you have?
So they're just, I don't know. It's weird. All right. Getting older. Anybody? All of us. Getting older is all in your head.
Yes.
Actually, it's happening all over your body.
Oh.
But
And my back.
by looking at your brain, researchers can approximate how old you are. Or were, because you have to be dead when they look. The researchers who examined post-mortem brain samples from 480 individuals ranging in age from 16 to 106 found that the state of someone's glia cells is so consistent through the years that it can be used to predict someone's age to a somewhat good, decent degree of accuracy. Why they say the state of someone's glia cells is because they found that the glia cells, which are a support cell to the neurons, change over time. This is so they also the other flip of this is they discovered that the difference between an older brain and a younger brain isn't the number of neurons, that the number of neurons isn't that dramatic. And that, by the way, that is the absolute worst infographic ever created. I'm looking at this. I have no idea what that's supposed to mean. This infographic that's we've got up on the screen now. It make no sense to me. Make no sense. I looked at this and I went,
colors.
this is not helping me understand anything. Actually, I'm more confused by looking at this. But the fascinating part of this, the other part of that is fascinating. This is that they discovered the difference between the old brain and young brain. Neurons don't change that much. But it's the function, presence, and support of the glia cells that are there to support the health of the neuron. This is what changes over time. It says here, we extensively characterized age-altered gene expression changes across 10 human brain regions and found that, in fact, glial cells experience bigger changes than neurons, says Jerne Jule, neurologist at the Francis Crick Institute and University College London, who led the study. There's quite a bit of regional information that will be of interest to different people. For example, some will notice a very unique pattern of astrocyte-specific changes in the substrantia nigra that we provide a lot of data that I have no idea what he's talking about there. But there are 3 types of glial cells, each providing different kinds of support to neurons. Oligodendrocytes, that they say here, insulate the neuron. Microglia cells act as an immune cell, and the astrocytes, which help the neuron metabolism and detoxification efforts, right? Based on analysis of human brain tissue samples, primarily they did this from the UK Brain Expression Consortium, which sounds like a bunch of gravediggers, doesn't it? UK Brain Expression Consortium. You need a brain, we'll provide one.
I will express it.
Express service of brains. This one's name was Abby. Abby what? Abby Norman. Don't worry, it'll be fine. Researchers showed that the astrocytes and the oligodendrocytes shift their regional gene expression patterns upon aging, particularly in the hippocampus and the substrata nigra, important brain regions for memory and movement, respectively, while the expression of microglia specific genes increases in all brain regions.
Basically, the microglia, which is working on the immune system, is the immune system portion. It's like, there's a lot of work to do. You're getting
right,
old.
or is it
we
like
gotta
an
keep
over
down the inflammation? Oh my gosh!
But is it also possible? Like this is what we don't know because they just discovered this. Is that sort of like is it possible? It's sort of like when you have response that's too strong, could it be causing the problems? Like this is a whole thing though. When we've been talking about neural health, now knowing that it's the neurons themselves seem to still be there. It's the real functional problem of aging, cognitive issues and motor function issues and the like, memory issues, may actually be in that support system of glia that then might be able to, be delved into, and maybe that can be controlled in some way. Maybe that can be sustained for a longer period of time, and your glial health will lead to neural health, of course, but the neural health itself seems to already be there. So,
Got to support the structure.
Right.
Just
So
keep it healthy. It's interesting.
This can be huge, from everything from neurodegenerative disease issues to just any kind of aging issues. Now that we can sort of focus in a little bit better on what it is might be going on. And then my last story for the night was just sort of tied to this a little bit. The microglia, which in this study says are constantly scanning the brain to find and fix issues. It calls them the housekeepers of the brain. Mm-hmm. It's thought that they kind of what you had is what you had for your lifetime, and they just went to work. It says the microglia cells are renewed several times in a lifetime. So this is sort of interesting, and it sort of makes me think, like, based on the last study, how the microglia are the ones that seem to be showing up more and more in increased numbers throughout the brain. Now that we know that they're renewed several times, maybe there's more hangers-on, from before. Like maybe their numbers sort of actually are, they are increasing, but as they're renewing, maybe something about having too many of them could actually even be an issue here. But this is
so I can't
how are they
prolong my
involved
life
in
by
disease?
eating a bunch of glia, is what you're saying.
You can't. No, don't eat glia. No.
Okay.
brains.
Okay.
I don't know if that
brains.
does anything.
Okay.
It'd have to be human brains.
I just want
They'd
to make
have
sure
be
I understood
fresh.
properly. Okay, good.
Yeah.
So the
Okay,
microglia.
Express Consortium and
And then you'll be at risk for kuru. No,
Okay,
don't eat brains.
So eating microglia not going to get me to my 200th birthday.
No, but maybe there are, since these microglia, like neurons, the idea is that they're very long lived because they have to support all the things that you do your entire life, your memory, lifelong motor memory, your abilities. So the neurons in your brain have to exist for your life, whereas the things that support them and keep them healthy can die off and then come back and refresh. And so it's interesting, this study, that they can have a bunch of renewal cycles, and that these microglia actually keep going. And so maybe there is stuff that goes wrong with the microglia. Maybe they stop cleaning things up as well. They stop renewing in Alzheimer's disease. Maybe this is part of what's going on.
So in this other study, though, it says it also found that the number of microglial cells remains relatively unchanged from birth until aging, and is maintained by the spatial and temporal coupling of cell division and death. But which is slightly different story. And when they say aging, the other study did go to 106. They may have stopped at their aging at 70. They don't have that detailed here from the part that I'm looking at least from this little overview of it. But so now knowing that, didn't we used to think this about fat cells too? Like you were born with so many fat cells and that's what you have forever. And then it turns out, well, actually there's quite a bit of turnover. We're now learning this too of our microglia.
Yeah.
Absolutely. And in the chat room, Rob the Stressed says, as for eating brains, you should eat brains if you are a zombie. So I think that's probably well taken advice. And along those lines, a story came out this week. Some students at the Department of Physics and Astronomy at a university, they put together an analysis of how long it would take a zombie outbreak to kill off the human population, or if it was the University of Leicester. There it is. And they published this in a student-run journal called the Journal of Physics Special Topics. They discovered that if there's a 90% probability that if an uninfected person who met a zombie, that an uninfected person who met a zombie would get infected. So 90% chance that zombies would pass on their infection, 10% chance that they would not. Basically, they discovered that if humans are unable to defend themselves, all humans, humanity will die out in less than 100 days from a zombie outbreak.
Yeah, that checks out.
Which is with a population of 7.5 billion people in the world. With one zombie at day 0.
Because humans have to sleep and eat.
Yeah.
Well, I mean, the premises of this is that humans cannot defend themselves. Let me tell you.
But
if humans could not defend themselves, cats would kill us before zombies would kill us.
Right.
This is like an unfair situation.
Yeah. So then they looked at it again and they're like, okay, if people can defend themselves, then it takes us a period of time for the infection to spread, but then humanity will prevail if humans can actually. Yeah. Zombies disappear completely over something like 6 years.
Double tap.
6 years.
Yeah.
That is too long with zombies. I am not into that.
Be fighting zombies. The zombie apocalypse will take about 6 years. Yeah.
I'm gonna go to Mars if there's.
I don't think zombies. I've never been afraid of zombies, and they're just too slow moving. It seems like most of their brain function, outside of the desire to eat brains, has ceased. I feel like it's not too hard to outsmart a zombie.
Okay, Justin, I'll be sure to call you when I can't sleep tonight because we've had this conversation then.
That's right.
We do know it would be a major metropolitan area. Something.
Let's talk about science.
A place that had sort of bottlenecks.
Let's talk about the appendix.
In and out of it.
Let's talk about the appendix.
Let's talk about the appendix.
Let's cut out this conversation so we can talk about the appendix.
We're going to talk about the appendix. You know, that thing that gets infected every once in a while and you have to go to the hospital and have it removed. The appendix. Yeah. Yes. Appendicitis from its inflammation is, that's terrible. Why does this happen? Why do we have this vestigial organ? What is the appendix even good for? People?
Nothing.
Well, it's been posited that maybe it is a repository for bacteria, and that beneficial gut bacteria hang out in the appendix. And so, and so a researcher, Heather Smith, associate professor at Midwestern University, Arizona College of Osteopathic Medicine, is studying the evolution of the appendix across mammals. She has looked for the presence or absence of an appendix in 533 mammal species. And then they put it onto a phylogeny. I know you love the phylogenies, Blair, to see basically how the appendix evolved over time, why some have it and why others don't. They found the appendix has independently evolved over 30 times. And when organisms get an appendix, they don't lose it. So it probably has an adaptive purpose. It's
So
not
it's not a
detri-
vestigial.
It's not vestigial. It's not
It's
detrimental.
convergent.
It is convergent evolution that it has
Wow.
independently evolved over 30 times. Yes, convergent evolution within mammals. And they also find the species with an appendix have higher average concentrations of immune tissue in the cecum portion of their large intestine. And so it's because of this correlation, it's also supporting the idea that the appendix might have some kind of job in the immune system, that it might be a secondary immune organ, potentially helping being a safe house for gut bacteria.
It protects you from zombification.
It's like a bacteria frat house.
The only people who become zombies are those who have had their appendix removed.
Yes, and so they've
correlation.
also concluded that the appendix doesn't evolve all by itself. You have to have a certain shape intestine, a ceca. It has to be tapering or spiral shaped for you to get an appendix, and so it doesn't evolve on its own. It is part of a larger cecoappendicular complex.
So
So it's something it's evolving in conjunction with the cecum, the small intestine, probably with this immune tissue and for some immune purpose. So very
does
interesting.
this mean that when people get their appendix out in future, it might be coupled with some sort of treatment to replace what you're losing?
Potentially, yeah, if we can figure out exactly what the appendix. I mean, people who get their appendix removed don't necessarily have a hard time of things, right? But there is a question that is the appendix going to help people bounce back from certain gastric insults? Is it? I don't know. We don't know.
Or is there something going on? Yeah, that we've never noticed the
We haven't
correlation
noticed.
before.
Yeah.
Absolutely.
Right. So fascinating. And moving into my last stories here from the appendix, I'm going to take a jump into superheroes.
Ooh.
Superheroes.
Appendix man.
Right? Superheroes, they do good deeds. They save people, but they also do a lot of punching and kicking, and they can be really aggressive, and sometimes they use weapons. And according to a study, when kids watch programs or get into superhero culture, a lot of kids don't actually take the good side of superherodom to heart and have that be what they identify with. Most of them identify with the aggression. And so researcher Sarah Coyne, who last year or so studied princess culture, she's now moved on to superheroes.
I would love to hear about that.
Yeah, she published in the Journal of Abnormal Child Psychology on this effect, in which on the playground, basically, kids would be less likely to respond to another child in need on the playground, even if they were into superheroes. Coyne says, though, don't worry about not allowing your kids to see superheroes. She says, moderation, have your kids involved in all sorts of activities, and just have superheroes be one of many, many things that they like to do and engage with. And so some aspects of this study that I thought were interesting were some of the things that the kids said. So they looked at 240 children, and the parents responded about how engaged in superhero culture the kids were. Parents were asked how often their kids watched superhero media, how much they identified with different heroes. And then the kids were interviewed, and they were asked to identify superheroes, identify their favorite, and then explain why they liked the superhero the best. So responses included merchandise, so lots of superhero clothes and toys and stuff, image, and interpersonal characteristics. And so these those who specified characteristics in superheroes, 10% noted some defending ability. Only 10% noted the defending ability. Quote from a child, because he shoots webs and he saves people. 20% of the kids associated the hero with violent stuff. He's big and he can punch. Some were mild. Others were really, really aggressive. One kid was quoted as saying, because he can smash and destroy everything and doesn't care, because he's a big bully. Mm-hmm. Another child stated that Captain America was his favorite because he can kill.
So I'm wondering about chicken versus egg scenario here. So do these kids consume more superhero stuff because they already have these violent sides to them,
right?
Or are the superheroes breeding this violence? I would say it's probably the other way. It's probably that the superheroes appeal to this side of them that they're figuring out. They're kids, they're animals. They're trying to figure out how to control impulses and figure out how to not do what biologically we're so well tuned to do, which is fight.
Okay, so let me do a really quick clarification on everything. Because I see all, I know all when it comes to this.
Oh boy.
When I was a kid, being into superheroes heavily meant reading a comic book. And in the comic books actually didn't have a heck of a lot of action.
That's after you can read. That's when you're old enough to read. That's when you're older, and we're talking about younger kids.
Sure.
So but if you're
right, and this is sort of my point. This is my point. And so, like,
it's TV's fault.
I liked Spider-Man because Spider-Man was really snarky. He had snarky opinions on everything. He got into fights, and he had like this crosstalk he'd be doing, and he had a lot of self-doubt, and he'd be like, "Oh my gosh, that really hurt." Like, you know, he was like, and sort of like very, you know, they wrote through all of his fears about fighting a bad guy. Then he has to fight him and he can't back down. He's being snarky and humorous about it when he's talking to the guy, but in his head, he's like, this is going to hurt. Right. That was a nuanced character. What the kids now have, it's all action movies. And you could, I think, easily take a superhero out of there and just put a ninja, cowboy with a gun, whatever it is. And it's an action movie. It has nothing. There was, when you were, you know, when the superhero portion of these things were created, a lot of it by I'll use Marvel's Stan Lee, he was looking for nuanced characters, some part role model, some part freak that didn't fit into the world. And that was like a home, like if you could read these guys, and these guys aren't perfect, but look at what they're doing. They've found a way to be heroes in the world, even though they don't fit into it, even though they don't quite belong to it. So that was like a hugely different thing than somebody who's just smashing stuff on TV and has this ability, this power to break stuff. And I think that's a big problem. I think superheroes, as we think of them, as us, I mean, older-ish people, the whole superhero, truth, justice, the American way for the, you know, good of society portion of it is going to be lost, because in an hour and a half of everything exploding on screen in one big car chase, none of that's going to be communicated. So the superhero doesn't really have so much of the hero involved, just the super ability to break stuff.
Well, but so it does come down to, making sure that whatever your kid is consuming, it's balanced.
Right.
If they love Star Wars, that's great. They can watch Star Wars, but they should also watch some other stuff.
Right.
And
don't get so focused on one
Doctor
particular
Who,
thing.
as Ed from
And one big part of this is, like, you know, I said 10% noted positive aspects, 20% noted aggressive, and 70% of the other characteristics comments were benign. So just he's big and strong, he's cool, it can fly, you know, that whatever superhero it was could just do something neat, or could just do something.
And that kid can't relate to the superhero because he doesn't know the superhero also wet his bed like the kid's been fighting. And if he knew that, he'd be like, he's the most awesome guy ever. He like goes out there and he also overcame bedwetting. Like, whatever it is. Like,
it's just
all that, like, without the comic book portion of it, superheroes, I think, are a dying breed.
Yeah. So we don't want to take this study too far,
Right.
but it's, it's like I said, we don't need to get rid of superheroes for kids. It's moderation.
We just
Kids
need to make them
most kids
read.
are pretty cool.
It's fine. They can consume things. They can like certain elements. It's up to us as humans to shape other humans.
That's
Yeah,
right.
and make superhero movies more boring. Do a whole lot of backstory and talking
Yeah.
where you get to learn that the character is flawed before he goes out and just smashes
And
stuff.
smashes things, right?
Yeah.
And we did our predictions show last week, but there's a few astronomers who have made a prediction. They presented their findings at the American Astronomical Society's meeting in Texas last week. They've been studying the binary star system KIC 9832227, and these binary stars, these two stars that are orbiting with each other, they're getting closer and closer, closer and closer, closer and closer. And this system that's 1,800 light years away, they predict is going to have the stars smash into each other in about 5 years. So in 2022, we should be able to see a red nova in the sky. It'll be red in color, and it will be bright enough that it should be visible from without a telescope from most urban areas. So it'll be just a light in the sky. It's not going to be crazy, but it's going to be a new little star that we can see.
Cool.
2022. Neat. Yeah, magnitude 2.0.
2.0.
Yes. Anyway, yeah, and then it'll fade away. But anyway, very exciting. Maybe 5 years from now, we'll have something to look at in the sky.
Excellent.
Blair, tell me a story.
Oh, I didn't tell
Close
you
up
about
the
my
show.
armpits yet.
No.
So not my armpits. Orchids have armpits? No. Orchids have been found to mimic body odor.
I know. Ew.
The Department of Biology at the University of Washington, Dr. Jeff Riffle there has been squashing through the field looking at orchids, and the orchid Platanthera obtusata has taken advantage of mosquitoes. Mosquitoes are attracted to these orchids for smelling like, as they so eloquently put it, a sweaty gym sock or an armpit. And these stinky, stinky orchids, they were found to have these pollen sacs over these people's heads as they were trudging through. And on examination, when they collected a bunch of these sacs, they discovered that they were quite smelly, looked closer at the chemical signals involved, and found that they were pretty similar to human stench, let's call it, and that may be, in fact, to attract the mosquitoes to have an unlikely pollinator. So we talk about mosquitoes being useless all the time and having no function. If they're pollinators, that kind of ruins all of that.
Oh, yeah. So if we get rid of mosquito, we also have to lose plants that smell like BO.
Yeah.
Oh, yeah. Not worth it. Not worth it.
But this is actually some of the first demonstrations of how mosquito nervous systems process sensory information. And what I found especially interesting is, after looking at the chemical signatures behind these orchids and how the mosquitoes responded in their nervous system, they looked at the mosquito's brain's response to DEET to see what would happen. And it activated all sorts of neural pathways, leading the team to conclude that DEET might be, quote, really confusing to a mosquito, which makes me think, now if we know a little bit more about the neural pathways of mosquitoes and what they're attracted to, we could actually fix things like DEET to be more effective, potentially.
Attracted to
cool.
stinky armpit flowers and cows. Didn't we learn that?
Yeah. So these orchids smell like sweat, and we could use that for science.
Oh, and on that note, we have come to the end of the show. Stinky armpit science. Yes. All right, everyone out there. Thank you so much for joining us. We've made it to the end of another hour and a half. And I would like to take this opportunity to remind you once again about our TWIS calendars. Go to twis.org if you are interested in finding out more. Facebook.com/This Week in Science to find out about our January 19th show at the Cal Academy of Sciences in San Francisco. And shout outs to all our science homies in our chat room. I see you there, writing your words, being humorous, being interested in our science. I see you, chat room. Thank you. And I'd also like to say thank you to our Patreon sponsors. Thank you to Chris Clark, Paul Disney, G. Burton Lattimore, John Ratnaswamy, Richard Onimus, Byron Lee, E.O., Jared Lysette, Kevin Parachan, Andy Groh, Keith Corsell, Jake Jones, Ulysses Adkins, Derek Nickel, Dave Friedel, James Randall, Eric Schwalb, Bob Calder, Mark Mazzaros, Ed Dyer, Trainer 84, Brian Hedrick, Layla, Marshall Clark, Charlene Henry, Larry Garcia, Randy Mazuka, Tony Steele, Gerald Onyago, Steve DeBell, XV, Daryl Lambert, Haroon Sarang, Melissa Mosley, Alex Wilson, Jason Schneiderman, Greg Guthman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts, Richard Porter, Rodney, David Wiley, Robert Aston, Aurora Lee, Bill Kersey, Ben Rothig, Darwin Hannon, Rudy Garcia, Nick Abbasi, Felix Alvarez, Cosmic Gypsy, Brian Hone, Orly Radio, Brian Condren, Marked, Nathan Greco, Hexator, Deborah Smith, Mitch Neves, Flying Out, John Crocker, Christopher Dreyer, Sylvan Westby, Artyom, Shu Wadis, David Wilkinson, Steve Mashimsky, Rick Ramus, Gary Swinsberg, Phil Nadeau, Braxton Howard, Salgood Sam, Matt Sutter, Emma Grenier, Philip Shane, James Dobskin, Kurt Larson, Stefan Insom, Michael George, Russell Jensen, Mountain Sloth, Jim Drapeau, John Maloney, Jason Olds, James Paul West, Alec Doty, Illuma Lama, Joe Wheeler, Dougal Campbell, Craig Porter, Adam Mishkan, Aaron Luthen, Meg Marjorie, Paul Stanton, David Simmerly, Tyler Harrison, Colombo Ahmed. This list keeps getting longer and longer, and I thank you for being a part of it. Thank you, all the new names. Love putting your names in there. Love reading all the names every week. Thank you for your support on Patreon. And if you are interested in finding out more, please go to patreon.com/This Week in Science. Remember that you can help us out just by telling your friends about TWIS. And on next week's show, once again, we will be back at 8 p.m. Pacific time on Wednesday, twis.org/live. You can watch and join the chat room. But if that doesn't work for you, you can find our past episodes at twis.org/YouTube or just twis.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can look for TWIS, the number 4 Droid app in the Android marketplace. Or simply This Week in Science and anything Apple marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org.
What?
www.twis.org.
One more time.
That's www.twis.org, where you can also make comments and start conversations with the hosts and other listeners.
Or you can contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. You can just be sure to put TWIS, TWIS, somewhere in your subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIScience, at Dr. Kiki, at JacksonFly, at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week. Where? That's right here. And we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
This Week in Science. It's the after show now, everybody. This is the part where we're after the show. It's the after show.
I'm gonna turn my space heater on. It's cold.
Oh, hush. It's fine. I'm sure you're cold. I'm sure you're cold. My friends have been text messaging. There's like this long list of
I'm not wearing enough layers.
text messages. How it's probably what, 50s there?
Well, in my house, it's like 62 right now. It's not that cold, but it's just cold for inside the house, you know?
Yeah, cold for the house.
Yeah. Strengths, I didn't talk about the deer molesting. It was funny, it was interesting, I guess, but it wasn't much of a science story. It's something they'd reported on before. It was a repeat of something they had seen a few years ago. I guess it was kind of interesting, but there wasn't a lot to talk about there. It's pretty self-explanatory. Monkey meets deer. Deer stands there a while. Deer runs away. Deer
Oh dear.
licks a substance off of its back. That's pretty much it.
Oh dear.
Yeah. Oh dear.
Oh dear. Hi, Chai. Chichi Rodriguez. Woo. Oh, get some good food, Fada. Thank you for watching. What was another story I didn't talk about this week? Oh, the Alzheimer's teeth.
Pardon?
Alzheimer's teeth. Researchers are using some kind of Alzheimer's drug to potentially regrow teeth or to fix teeth, to strengthen teeth. Yeah. Interesting. Interesting.
I'm really excited maybe next week at Sketchfest to talk about how scheduling leisure activities makes them no longer leisure activities.
That'll be good. I know I need to get you a calendar, Fata. I've got one for you.
Oh, did we send one to Patrick yet?
No, I have to send one to Patrick and to
Okay.
Fata.
Okay.
Sending of the calendars must happen.
How many do we
I
have
need to send
left?
one to Justin.
Oh yeah. Or you could just
Mhm.
bring one next week, right?
That's
to Justin.
what's gonna happen. I'm
Yeah.
gonna bring one and give it to
Y-
Justin.
If there's not that many left,
And,
maybe just bring them all.
well, there's enough.
Okay.
I'm
Bring
gonna be
some.
staying in the Bay Area for 4 days.
Ooh, fun.
Yeah, I'm gonna leave
Yeah.
on Sunday morning, Sunday
Okay.
afternoon.
Yeah, bring some, and I bet I can sell more. We can sell some at the show, maybe.
Maybe, possibly. That would be awesome. Yeah, we'll see how many are left, and I'll just bring them. Maybe I'll bring a few.
Great.
Bring a few. Yeah, we're getting down there. Getting down there, almost ish.
Good.
Yeah, there were a few more orders this week, which is good. So
Excellent.
yeah, we'll see. We might be like scraping the bottom of the barrel.
Yes.
Which is great.
I am kind of regretting my decision when we were like, should we do a show the day before we do the Sketchfest show? And we were all like, yeah, it'll be fine. And now I'm like,
Why? No, it'll be good.
ah.
It will be good.
Thankfully, I did just find my printer.
Oh, that's good.
So print stories. This is going to be exciting. I'm going to have to actually. I found it. Now I've got to actually hook it up. But
Oh, I have to. The Alzheimer's teeth. That's a really good question, Dictel. Was this the first time Kai got to play in the snow? No, we've had some other snow before, and we've gone to the snow before. We've gone sledding. It's fun.
Nice.
So, but today Kai got some really good play in the snow in.
Nice.
We'll have some more play in the snow tomorrow because there's no school tomorrow because
boy.
Portland has 2 snow plows.
Yes.
We maybe bought our 3rd yesterday. Maybe. I think maybe the city bought a 3rd one. That's gonna be exciting.
I've never made a snowman.
What?
Yeah.
That's like you have to make a snowman if you're in the
I've been to
snow.
the snow like a handful of times, and it was very like, okay, get our butts up there. Like, snowboard. Okay, get our butts home. There was no dilly-dallying.
No dilly-dally.
oh,
You and the
euphemism.
dilly-dallying. What is
That
falling? What just
euphemism
happened?
returns. No dilly-dallying on
No
snowboarding trips.
dilly-dallying.
I dropped dice everywhere.
Why are you playing with dice?
I'm not. I dropped
She's just a degenerate gambler, and we can't see, but there's a
So,
whole
that's right.
dice
Blair is
game
so
going on the side
California.
over there.
Look, this one
I know,
has
there's a dice
a
game. I
raccoon
know.
on it.
Oh, those aren't. Oh, yeah, those are story cubes. So those aren't regular dice.
I don't know what those are.
You don't know
cubes.
story cubes?
No.
Story cubes are so fun. I play them with Kai all the time. You throw them out, and then whichever cubes come up, then you use them to make a story. So you can use them to cue aspects of a story.
Okay, well, they also have
Oh,
numbers,
that's clever.
though.
I like that activity.
They do also
Clever.
have numbers. And then there's this one that just has X and then, like, exponents.
Oh, I don't know what that's for. Maybe it's
I don't
not
either.
then. It's something
But,
else.
It's story time for mathematicians. It's just
And then
I know.
have,
Kiki, story
um,
cubes are fun.
dice with
Cinque.
it's
Cinq.
French.
1, 2, 3, 4, 5, 6,
And then
7,
I have one with
8,
Hebrew on it,
10.
which
Hebrew.
they're letters. I don't know. And then I have
What
dice.
kind of Jew are you?
No, but they're letters. They're not. Anyway.
They're not words.
But they're not numbers, which I'm saying is weird. There's dice inside dice.
Why do you have so many different die?
Because I used to collect dice. So
You did?
yeah. So then it was
You
one
and
of those
your
things
collections,
where like
man.
somebody was like, "Oh, Blair collects dice. I'll get her dice." So then like whenever people went anywhere, they brought me back dice. But then there's the giant foam dice with animals on them, of course.
I went through that for a phase for a little bit. I had, for some reason,
Right,
people
Dungeon and
all thought
Dragons,
I
that's what
liked
that was.
like book place markers. So I was getting all kinds of different
Bookends?
not bookends, but like the place markers, like so you keep your place in the book. What do you call it? Bookmarker, right?
Bookmarks?
Yeah, and I got a couple of them were pretty
What?
cool.
Did you just forget the word bookmark?
Yeah, I haven't read a book in a long time. That's why I was like, you have to stop giving me a book. I don't read books that much anymore.
Dang.
But I
I
have my
had some-
D&D dice right
Some
here.
cool ones. I don't know where they are. My kids stick them, but there were a couple that were shaped like little rugs, and they were supposedly like marked off of fame. Like one of them is supposedly like Freud's rug, and then you would like, yeah, that's cool.
Freud's rug.
Yeah, it was a bookmarker I had. It's a whole thing.
It is a whole thing.
Deep into the book marker collection. Stick.
Get into the bookmarker collection. Ooh, I'm going to let Dictel join Ccience with a Twist. I think we'll do that. I can do that.
What?
I did it. I'm letting people join things on
Cool.
Facebook. Bye, Kiki.
So yes, I dropped
Turtles.
something onto the...
I'm still dealing with no snowman build. I know she's playing with dice and she's never built a snowman.
I never built a snowman. I've never like stayed overnight in a place with snow. I feel like if that had happened, I would have ended up building a snowman. But it was always a day trip. It was always like get there, get things done, and then we're tired, so let's just go home, sort of thing. Like,
always so tired.
Yeah. So at some point, I'll have to get myself to somewhere with snow in a cabin and just frolic for several days. Maybe next winter. You know I would frolic in the snow.
Oh, yeah.
It's actually, I have to show you the snowsuit I want to buy.
You're going to buy a snowsuit? Of course you are.
When's the last time you went to... Never mind.
One of these.
I've seen these. I know what you're doing.
Yeah.
I know what you're bringing up.
Woo! There's one in particular I want, but they're sold out. So I'm definitely... That's a problem. That's a problem.
Oh my goodness.
The one I really want is sold out, which is so very sad.
Snow 4 to 6 months of the year. Yeah, I'm not interested in that. That's a little bit too much. There's a bit too much happening here in Portland at the moment. I didn't sign up for all this snow. I didn't think Portland was such a snowy place. Whoa, this is weird. Somebody made a kitchen backsplash out of LEDs. That would get obnoxious really fast.
Right, yeah. Oh, gosh.
Oh, my.
Oh, no.
It's all LEDs.
That's terrible.
Oh, I hate it.
It's a little bit too much. I mean, I guess you can.
It's a fake aquarium. Oh, and
Yeah.
now it's a waterfall.
It's a waterfall. It's pretty. That's not pretty.
Just because you can.
It's
folks,
lava. Your
doesn't
kitchen
mean
is
you
hot.
should.
Your kitchen is so hot. Ooh, now it's like bubbles. Now you're in a bubbly percolator. Oh, it's a shark. It's an aquarium. Look at that. It's a shark, Blair.
Yeah, shark, 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 shark.
Oh, I like that. That's acceptable. But seriously, there's too much light going on. Oh my God, now it's like a hockey game.
It is. Oh, gosh. That's kind of funny.
That's funny. I don't need that. You never miss the hockey game again.
I wonder how many things got burned.
Can't even. That would just be. I'm gonna. It's too much. Too much LEDs there. Go away. I'm gonna make it go away. All right. So next week we're gonna bring our funnier side stories for Thursday.
Yes.
Information for Thursday.
When do you get over here?
Email.
In the morning on
I get
Thursday?
into town. That's a good question. Nope, wrong thing. What am I doing? I think I closed my calendar. Midday Thursday, I think. And then I think our call time is 5:30 p.m.
Okay.
at the Academy of Sciences.
Okay. Well, send me your flight info, because depending on the timing, if it is midday, I might be able to pick you up.
Okay. That sounds good.
Because I told work I was doing a half day.
Okay. Awesome. And where's Justin?
I'm here.
You're there. Where's your picture? What are you doing? Picking your
I'm
nose or something?
Yeah, hang on.
Justin, when are you getting to San Francisco on Thursday?
Yeah, when are you coming?
What time would you like me to be there?
I don't know. I guess it depends on when Kiki's flight is.
Okay. We'll figure that out.
We'll group text. We'll group text it out.
Yeah. I mean, the soonest I'd be leaving here is probably about 1 o'clock.
Okay, then leave there at 1 o'clock
Sure.
for sure.
Yeah.
Oh,
Yeah, one would be good.
okay. That would get me
so
like
I
3-ish.
should arrive about 2 o'clock.
We'll pick you up.
on Thursday. It's about 2 o'clock on Thursday.
SFO? You're flying into SFO?
Mm-hmm.
Okay, I'll pick you up.
Okay. Awesome. That'd be great.
And
Yeah,
then
it's a pretty easy drive from the zoo over to the airport. Actually,
yeah, it's only like 20 minutes. It's fine. Yeah, I'll pick you up and then we'll swing by my house where I'll grab my laptop because we'll need that, right?
Mhm.
Probably. And then we can head over that way and maybe camp out at a restaurant or something to go over stuff, or in
Yeah.
the academy or whatever we want.
Mhm.
I'm going to
Sounds
pick up
great.
my badge tomorrow, I think.
Okay, because I sent you all the information. You
yes.
got that, right?
Yes.
So when will the episode start on Thursday? That is a wonderful question. I will find out. I have
Yeah,
no
last time I
idea.
saw that we will be on some time between 6 p.m. and 10 p.m.
Yeah, actually, I'm going to. Our call time is 5:30, so I'm going to email my dude now. Okay, great. What time is our performance?
Are they streaming it?
It's if we can make it happen.
Oh, okay. So
So-
depending
so
on the
no.
Wi-Fi. So
Depends on
probably
the
not.
Wi-Fi. Depends on it depends on a lot of factors. So I'm hoping we can make it happen, but
Do you know
may or may
which?
not be able to, because I have one laptop I can bring. You'll have your laptop, and
Right.
then yes, and I'm going to bring my audio recording device, so there will be a local recording of the show.
And
Yeah.
do you remember which venue location in the academy we're at? Was it on the terrace? Or were we in Africa Hall? I don't remember.
Did it say where we are?
Maybe I just know there are 3 spots in the academy where people
Yeah.
are performing, and I wasn't sure which one. So I know I've done events for other things at the academy where I've had to use Wi-Fi, and so I know there's some spots where Wi-Fi is
Yeah.
better than others.
Okay, so I'm just asking what time. I don't think they've told us anything about where our performance is yet.
That's fine. So yeah, our Wi-Fi quality will
Unless I
just
just
depend on
missed
that.
Yeah. I could have also just missed it. I got a lot of things running around my head lately.
Yeah. Lots going on.
Sometimes I just miss things. I'll do nightlife. Let's see what emails I have. Yeah, somewhere in the 6 to 10 p.m. range so far. And that's all I know.
Okay.
Call time is 5:30. That's all I know.
Cool. Great. Exciting stuff.
Ba-dum-bum. Yeah, so next week we'll do a regular show. I don't know if we will have an interview next week or not. I'm trying. Yeah, so Hot Rod, if I can periscope it, so I'm going to try and do whatever I can to make a broadcast happen. I'm going to try and do it through the normal YouTube channel, so it goes out through our website the way that things normally happen. But if we are limited by computers and technology and Wi-Fi and all that kind of stuff, absolutely, I might just turn it into a live periscope. I might just do that, and I think that could be cool.
Okay.
And it's possible also if I just turn on if the Wi-Fi is acceptable, just turn on a Hangout on my computer and then just ignore it and put my screen in front of it and just do all my stuff that I normally do with my computer. Yeah, it's only a 40-minute show, so
and I have a soundboard. I'll just keep dropping things. I have a soundboard if we want it, but I bet they're going to be taking care of all that, right?
yeah, they're doing AV.
Okay.
Yeah.
Okay.
They're doing the AV, but we might have to do our own music. So like we did, like we've done before.
Yeah, that's fine.
Yeah.
I can do that from my laptop. You can do it from yours. We can figure something out.
Yeah, and hopefully we'll get video output so we can put images up for the stories that we're talking about also.
Yep.
Yeah.
Cool.
Yeah. So what aspect of the show what clip did you share? Do you remember what you shared when you did the application?
When did I apply? Before Baltimore, right? Or after Baltimore?
No, it was like in August or September.
Okay. Yeah. So then I just sent them the link to the New York City show. They asked for a 5-minute clip, and I was like, hey, I don't have a 5-minute clip, but I do have this fully produced entire show that we did in New York.
Nice. Okay.
Fair enough.
Awesome. All right.
Because
Perfect.
I was like, I don't have the time or abilities to edit a reel right now. So here.
Hot Rod, you're following me.
I think I did that. And then I also linked one of the shorts. I think I used one of the shorts also.
Oh, cool. Okay.
I don't remember which short.
Oh, now she's yawning too.
I got everybody. Sorry.
Everybody's now yawning.
Yeah, which short did I send them?
That
I have no
part
idea.
Doesn't matter. We're in. Now what matters is what we're gonna do live.
Yeah. Yeah, I mostly just leaned on the New York show because I thought it was like, just very a fun atmosphere. You could hear the crowd laughing sometimes, like whatever. It was fine. So it was very representative of the show we would actually be giving. So
yeah.
So, but Blair, what you have to bring has to be enough to get you on Chris Hardwick's midnight show,
right? Yeah.
Yeah, that or I just I want to be on Comedy Bang Bang.
Okay, so there you go. So just be really.
I feel like I could be on Doug Loves Movies pretty easily.
Just be really funny.
Right.
That's all you got to do, and not screw up or be nervous.
Justin,
yeah, don't be nervous.
Thanks.
Don't screw up at all.
Thanks for the tip.
Yeah, just don't.
No, see, I'm going to go get my free massage at the Performers Lounge, and I'm gonna find a way to get my massage right next to someone famous, like your Chris Hart.
Oh, that's a good idea.
Strike up a convo.
This is a good plan. Yeah.
I'm going to be at the open bar for the pre-show.
Yeah.
Is it
Well,
open
that
bar?
sounds about par
No.
for the course. There's not. No, you got to pay
No.
for it.
All right, then I have no pre-show plan.
I don't know. Maybe there's stuff in the green room.
Oh dear.
We don't know.
There could be, but yeah, probably not.
I know. Did you put anything in your rider, like blue M&Ms or?
No. Nope.
A limo at 70 degrees.
What do you know? Do you know the story behind the green M&Ms or whatever?
Yeah,
it was?
it's so that they knew whether they were reading it or not.
Yeah, whether
Yeah.
they were paying attention to detail.
Yeah, Ed, what happens in the massage room stays in the massage room.
Oh dear. Like a massage.
Yeah.
Mm-hmm.
Yes. I want to go to bed, you guys. I'm real tired.
It's late night. I'm a bit tired also.
Really not that late, but I am also of the yawning now.
I know. I'm of the I could go read a book. I could go read a book persuasion.
I want to lay down.
So while we sleep, let's all come up with something to turn the show into a dynamic comedy event for
Well,
the
I already
Sketchfest.
picked some stories potentially for next week.
Okay. All right.
Like, scheduling my leisure time.
If you're
That's
good with
fine.
that, if you think that's enough, go to bed now. Don't stay up researching for the pure gold.
Oh, I'm sorry,
that you
Justin.
can
Are you telling me about how to prepare for the show? Is that something that you're telling me how to do right now?
No, I'm just saying if you're happy and satisfied with the little bit of, content that you've got, you think it's going to be good enough, by all means, just bring that. I'm sure it'll be fine. Don't spend too much time like working it, getting it really refined so that sleep while you can, why not? Why not? It's a long life, and nobody ever looked back and said, I wish I hadn't slept as much. People wish they'd gotten more sleep, is what they wish with their lives. Looking back.
How else am I going to get to live to be 200 if I don't get any sleep, Justin?
Yeah. Or you could just sleep less and live now, and it would be
No.
That's also a possibility. I'm actually not going to go to sleep. I've got a painting I'm working on over here.
Nice.
I'm going to go work on this painting for a little bit, and I'm going to fall asleep.
A flare
while
conversation in office space. That's funny.
Yeah.
So true. So true.
I want to lay down.
Oh my gosh. All right, everybody.
Are we supposed to have jokes though, or is this like a
No.
stand-up?
No.
Are we doing stand-up?
We're at the
no.
Science. We're at the Science place. We're
It's
at the.
We're doing our podcast. We are doing our show. We're doing
But
a
the
live
California
show.
Science is doing a science show, right?
So SF Sketchfest is comedy, right? But it's a science. It's a very science themed event, and the sketch doesn't have to be funny. It can be us doing our show. Sometimes we can be funny. We're funny.
Like
sometimes.
all the comedians are going to be doing like fart jokes, and we're going to be the thing that has the science theme.
No, okay. So
it's a
for
science
example,
event. What we're going to be is a science event.
Things you should know. You know that podcast?
Okay.
They've done stuff at Sketchfest. Star Talk Live. That's actually how they're opening Sketchfest tomorrow night is with Star Talk Live with Eugene Merman and Bill Nye.
Yeah,
that's very fancy.
Yeah, so that's the look at the event and look at the other things performing our night. So there's 3 different halls where people are performing. In one hall, there's like straight up ridiculous, funny, not sciencey at all stuff. In one hall, there's sciencey stuff that's also funny. That's where we are. And then in another hall, there's like sketch comedy going on. So we're in the hall with sciencey stuff that's also funny.
That can be funny sometimes. So, but we don't have to come up with a sketch. We don't have to come up with jokes. We don't have to come up with
It
stuff.
feels like we should be doing sketch comedy, is all I'm saying.
No.
Based
we're
on the
not.
name of the.
We're not doing sketch comedy.
No.
We're doing our show. We weren't asked to come. We applied and we weren't accepted based on the potential for our ability to do sketch comedy. It was
Right.
because our podcast was okay. They
Right.
liked
There's a. If I search the available Wi-Fi in my neighborhood here, that I'm getting
hardly
signal from.
strictly sketch humor. Nice.
There's one that's called Bill Y, the Science Fi,
Ha.
and it's not me. That's what's amazing about it. That's why I love Davis. Mm. There's also a Dumbledore 4 and a lot of just boring nothing.
Okay.
Not so boring.
Speaking of Dumbledore, I'm gonna go listen to Harry Potter on tape while I go to sleep.
Oh.
Don't laugh at her, Kiki.
I'm participating in a Harry Potter trivia contest in a couple of weeks.
Kiki,
So
don't laugh at her.
I'm brushing up.
You're mocking her. It's
I know,
not. That's
her
my
passion.
job.
Her passion.
That is my job. That is my.
No, I'm sorry.
You're
I'm
stealing
brushing
my
up
shit.
for the trivia contest.
That's great.
That's
really
fantastic. It's good that you put your study time into that as opposed to.
I'm just imagining
You
that's great. I'm just imagining
Your
you like
shows and
falling, drifting off to sleep every night to the dulcet tones of Harry Potter.
I know.
It's great.
Who
You
wants
should
to hang
try
out with
Jim Dale
10-year-olds all day?
sometime.
Oh wait, that's what I do.
Jim Dale.
Yeah.
He's great.
Mhm.
He's an award-winning audiobook narrator.
Narrator.
He's
Narrator. You know. All right, I'm done.
What just happened? What did I miss?
HP. Listening to the HP. What- HP narrator. I'm gonna HP. Gonna narrate the
Strengths.
HP.
No. Not Stephen Fry. Jim Dale. Jim Dale. Stephen Fry. He's great. He is nowhere as good as Jim Dale's version. FYI. Just so everyone in our universe knows.
I know
I'm
it's
not
not
having-
toast.
I don't know what. I have no idea what you're talking about.
You have two choices when it comes to your Harry Potter on audio.
Yeah.
Mhm.
Jim Dale or Stephen Fry. And you would think Stephen Fry would be the way to go. Not the case.
Yeah.
Jim Dale.
You know what the choice in this house is going to be? Me reading it chapter by chapter to my son.
That's also good.
Because in this house, I'm an award-winning narrator.
Good. Good. Good.
My kids like I love when I read them books until they learn how to read. And then realize I'm not reading the book. I'm like making up my own story.
No.
But there's some really good stories out there now that, I've got other pictures that go with it, but yeah, they realize there's quite a bit of ad-libbing, Papa. I don't see any of that in the text. Sort of ruins the whole magic when they learn to read. Then they're like, I'm just going to read it myself. I want to know what's actually in it.
I want to know what's actually happening in the story, Papa.
Yeah.
Come on, Papa.
How come every story is a moral theme about children cleaning their rooms? I cut on to you, Papa. This is not what every story is about.
I know. I could record my own Harry Potter series. Good, right?
Yeah, you could record. I mean, that's the beauty of it.
Could
You can.
read it.
That's what you do when you read. Is you're self-narrating.
I know. Self-narrating. No. Yeah, switching off. Exactly. Identity 4. Yeah, reading books with parents before bed. Loved it. My dad used to read to me, and we did the same. We'd switch once I could read, switch off chapters. It's great.
Oh, that's pretty cool.
So much fun. Yeah. Oh, and then post it on YouTube. Haha. I'll have to get my choosing hat.
Your
Yeah.
what?
choosing hat, the hat,
The sorting hat?
the sorting hat, whatever. The hat that
God. Okay.
chooses and sorts.
Okay. Later, nerds. Later.
Hey, I will own that.
I'm out.
Hey, you know what? You know what? I'm surrounded by the snow, so let it go.
Nope.
Let it
Yeah.
go.
No,
I'll take
it's
my
a-
Harry Potter. You can take that.
No, first of all, my kids turned this into a rap. Let it go, let it go. Can't hold it back anymore. Let it go, let it go. Turn around and shut that door. Yeah,
There we
it's
go.
turned into a whole. No, you know what's amazing? And I've got to get this videoed at some point. I think I talked about this. My middle child, Satya, can rap out the entire Hamilton thing.
Oh,
Wow.
She's
That's
got
impressive.
like and she's always been really good at remembering lyrics and verse. Like she remembers all songs, but she can like perform, like she can do the whole show. And she does it where she's like plucking on a violin because that's all they've taught her how to do so far is pluck this thing. But she's plucking out like this rhythm on the violin while like doing the whole show. Like I haven't seen it or heard it, but I know it now. Like the whole.
That's good.
All right.
All right, you
All right,
guys.
okay,
Everybody
later, nerds.
out-
I'm gonna go read my Choose Your Own Adventure combination Star
yeah,
Wars.
you go watch
and Star
Doctor
Trek
Who,
universe
and
book.
I'm the nerd.
Yeah, he's gonna go watch Doctor Who. I'm gonna go read. I'm reading a book
Actually,
called Exploded
that's
View.
true.
It's good so
That's
far.
true. Now reflect on it, Blair. I'm gonna go watch Doctor Who, and you are the nerd. Reflect on that, because that just said something very important. I don't know what it is.
I don't know what it was
Good.
exactly. I don't know what your point
Say-
is there, Justin.
I don't have one, but it's felt good to say that. Say goodnight, Kiki.
Good night, Kiki.
Say goodnight,
Good night,
Blair.
Blair.
Say good night, Justin.
Good night, Justin. Good night, minions.
Good night, minions. We will see you.
We'll see you next-
again. Yeah,
Maybe twice.
maybe twice next week. That is the hope. Have a good week.